Welcome everyone to today's HPL seminar. Today we'll be hearing from Dr. Seong-Won Han. Seong-Won is currently a postdoctoral researcher here at the University of Calgary with Dr. Walter Herzog. Her research interests include Titan's contribution to muscle contraction and force generation, and cardiac muscle mechanics and physiology. She completed her PhD also with Walter Herzog in August of 2020. For her PhD, she specialized in knee joint biomechanics and studied changes in patellofemoral joint mechanics in the presence of quadriceps muscle imbalance. Prior to her PhD, Seong-Won completed her bachelor's and master's in the Department of Physical Education at Yonsei University in South Korea. Her master's thesis advisor, Dr. Lee, was actually one of Walter's students about 25 years ago. Beginning in January, Seong-Won will actually transition to the University of Münster in Germany for ongoing postdoc research work. In Germany, she plans to research the relationship between Titin gene, truncated Titin, and dilated cardiomyopathy. As a fun fact, Seong-Won is presenting today because at the end of last year, when she was the moderator of the HPL seminar, Walter told her she did a great job moderating, but she didn't present at all during the year. So she wanted to make sure she had the chance to present before leaving for Germany. So as a reminder, I will first turn it over to Seong-Won for her presentation, and then we will have a question and answer period to follow. And at that time, I'll remind everyone how to submit a question. Thank you for presenting today, Seong-Won, and you're welcome to start screen sharing whenever you're ready.
Okay, thanks, Emily, for the introduction. Can you see my screen and can you hear me well?
Yes.
Okay, perfect. So today I wanted to talk about a small project that I did in this summer with an undergrad students. So before I talk about force depression in cardiac muscle, let me explain about skeletal muscle and muscle force and what has been done previously. So let's start with the structural hierarchy of a skeletal muscle. A whole muscle shown in here is formed of fiber bundles, or it's called a vesicle, which are composed of muscle fibers or muscle cell. And this muscle fiber consists of bundles of myofibrils, shown in here. And this, it is actually visible under the light microscope. So this photo is what I took from one of my experiments. As you can see here that it has a distinct, clear, distinct striation pattern. And this repeated units in series, in this pattern is called the sarcomere, which is the basic contractile unit of a muscle. So if I simplify this structure of a sarcomere and image here in schematic, one sarcomere is bordered by the Z line, or this red bar here in both edge, as well as thin filament or actin filament, which is this white thin structure, also has the thick or this myosin filaments, which contains projections called myosin heads, shown as these tiny short bars around the myosin. So the sarcomere is known to produce force through the interaction between the myosin heads and the actin filaments at this specifically overlapped area that I colored in yellow. And this interaction allows the actin filament, which is again the white thin filaments, to slide toward to the center of the sarcomere, causing the sarcomere length to become shortened, thereby producing force along the myofibril. And this action is regarded as the cross-bridge theory. And since the force is generated by this interaction of the thin filament and myosin heads, the magnitude of the force is determined by this overlapped area. That is, the overall force from this sarcomere is dictated by the number of cross bridges that are able to bind to this actin filament. And since the length of this entire sarcomere from Z disc to the next Z disc determines the amount of the overlapped area of actin and myosin filaments, force output of the sarcomere, of one sarcomere, sorry, is dictated largely by its length. So this is summarized as a graph here, which shows a modified active force length relationship from the classic study done by Gordon-Huxley and Julian. So on the y-axis, force is shown in percentage, while the x-axis sarcomere length is shown in micron. It has two phases of ascending limb, as well as one plateau region at the top and a descending limb. What this graph tells us is that the amount of maximum active isometric force is dependent of our sarcomere length, as I mentioned previously. So here I show you the previous schematic image of sarcomere, which the length of sarcomere is at the longest length, having no overlapped area between actin and myosin filaments. Since there is no, since no cross bridges are able to form at this length, the maximum isometric force should be 0, shown as this red dot on the force length relationship. So the red dot is located at the longest length, having 0% of force. However, as the sarcomere gets shorter and therefore more cross bridges are able to form, the maximum isometric force increases. When the number of cross bridges is at its maximum, then the force reaches to 100%. Also, since there is a bare zone where of the myosin, which doesn't contain any myosin heads at all, if the sarcomere shortens at this length through this bare region, the force still stays at the maximum because there's no changes in the number of the cross bridges formed. But however, since starting this length of the sarcomere, if the sarcomere becomes shortened, the maximum force will now start decreasing because of the reduced number of cross bridges interrupted by the part of this actin filament, as you can see in these schematics. And then also, once the sarcomere length reaches to this length where it touches the myosin filaments, and if the sarcomere length is shorter from this point, then the force decreases much faster because the myosin filament is somehow deformed, and it's somehow also like a squashed. So the force starts decreasing faster, and then eventually reaches to 0% again when there is no cross bridges that could be formed at all. So this relationship has been established for a long time. I think it's like over half a century, and traditionally used in the textbook and the research as well. But what I'm going to talk about today is one of the aspects that cannot be fully explained by this classic study, and it is called residual force depression, or shortly force depression. Then what is force depression? So here I am showing you an exemplar sarcomere that I colored green. And let's say the length of the sarcomere, this one, is at 3 micron. If I activate this sarcomere maximally without length change or isometrically, this green sarcomere will generate about 60% of muscle force, shown as these green dots, based on this force length relationship. Now let's have another sarcomere, which now I colored red, and the length is given at longer than 3 micron, or the longer than this green one. But this time we activate this red sarcomere maximally first when it is given at the longer length. And then after a few seconds, we shorten the sarcomere during activation till the sarcomere length becomes 3 micron, which is now same as this green sarcomere. According to the cross-bridge theory and the force length relationship here, the force generated by this red sarcomere is supposed to be the same as previous example because the final length for both these green and red sarcomeres are identical, meaning that the overlapped area between actin and myosin heads are also the same. Therefore, the number of cross bridges for both sarcomeres should be the same. However, it has been systematically found that in skeletal muscle, the force of this red sarcomere is actually lower than what it was supposed to generate over this green dot. And this discrepancy of the two forces, the difference between this green dot and red dot, is defined as force depression. The one question that we might have is why? Like, why do we see this depressing result? So the short answer to this question is that we don't know yet. But there are a few suggested mechanisms explaining or trying to explain why the force is lower after active shortening compared to the purely isometric muscle force. And I would like to focus on one mechanism that has been well accepted. So let's focus on the structure of the sarcomere. Considering the fact that muscle force is generated through the interaction of the actin filament and myosin heads, trying to bind to the actin filament. And when we see the lower force in this red sarcomere than this green one, there are two possible ways to think. First, maybe myosin heads, which again shown in this short bar here, generates lower force per one cross bridge in this red sarcomere compared to this green sarcomere. And the second possible way to think is maybe the thin filament or actin, the thin structure here, is not able to contribute to force generation proper way. So to explain a bit more detail, I just brought this red sarcomere back to its initial length, which was longer than the 3 micron. So if force depression was induced by the decreased force per one cross bridge here, or per one this short projection here, it means that the myosin heads at this specific area where I am now pointing at, or where the myosin heads have never bound to actin filaments yet, but will attach to actin filaments soon, generates lower force compared to the myosin heads that are in this area now that I am pointing at right now, or the area where this myosin heads already attached to this actin filament. However, this theory that the part of the myosin heads generates lower force compared to the other cross bridge doesn't seem to be the case. Instead, it seems to be more plausible that part of actin filament, specifically in this area, the actin filaments, or the I-band region, or that has not joined into the overlapped area yet, would be a cause of force depression. So what it means is that it was suggested that during activation and shortening of a sarcomere, the actin filament is exposed to a tetanic stress, and this stress strains the actin filament, especially that has not joined into the overlap area, but has little effect on the part of this actin where I colored, or where I still left the white color, where the cross bridges are already formed. So the proposed reason why the specific part of actin filament is strained and affected by the stress, while the other part is very little, is because this white part of actin filaments, where the cross bridges are already formed, has anchored to the myosin rod, while the actin filament in this area, or I-band, is relatively free. So when the strained actin filament now enter and goes toward the center of the sarcomere during shortening, this strained actin filaments would likely result in reduced chance for the myosin heads to attach to the actin filaments, then resulting in also reduced number of cross bridge in the sarcomere, therefore generating lower force in overall compared to this purely isometric force, or this green dot, even though the final length for both cases are identical. So this speculation or mechanism has been largely accepted and supported by many evidence, but we got one simple question on this. So if it is really true that the active components of a sarcomere, like actin and myosin heads, induce force depression, then we should be also able to see the same result when we use different types of muscle, which also have a similar sarcomeric structure in the muscle. So we started from investigating what muscle type would have similar structure of a sarcomere compared to the skeletal muscle. So here's a brief overview of the type of muscle. The first one is a skeletal muscle that I have been talking about. So when you look at the schematic of skeletal muscle, you can see that it has a striation pattern. I'm not sure if it is clear to see, but you can see like dark and light, dark and light, dark and light area is repeated, and it is repeated along the muscle fiber, meaning the sarcomeres are aligned in a row. So we search the further muscle types and see what muscle has a striation pattern similar to skeletal muscle. When we look at smooth muscle, which are usually found in the blood vessel and organs, we actually did not see any striation pattern at all along the muscle fiber. We found that smooth muscle actually does not have a sarcomere and rather have different alignment of the actin myosin filaments. When we looked at the properties of the cardiac muscle, we saw that there was a clear striation pattern along the muscle fiber. Here shown again, like dark and light area are repeated, meaning it has a similar sarcomeric structures that is there compared to the skeletal muscle. So we were curious if force depression is still shown in cardiac muscle. So we searched the literature and found that no one actually has done this work. So we decided to do a small quick project on measuring force depression using cardiac muscle. So the purpose of this study was to see if force depression exists in cardiac myofibrils. Since cardiac myofibril has a similar or almost identical structure of a sarcomere compared to skeletal muscle, we hypothesized that cardiac myofibrils will exhibit force depression. So in this study, we used New Zealand white rabbits. So heart was harvested from it, and after harvesting, heart was opened and dissected in order to get the left ventricle of the heart. So once it is harvested, it was cut into a small piece and stored in the freezer for 3 weeks. And after all skinning procedures is completed, we blend and homogenize this muscle in order to get a single myofibril from this isolated muscle, which looks like this. So as you can see here, single cardiac myofibril is mounted in our testing system. You can also see clear striation pattern there. The left end of the myofibril is fixed at the tip of the glass needle, where we could adjust the length of the sarcomere. And also the right end of the myofibril is attached to the left side of the cantilever. So in order to measure the force, we first measure the distance between the reference cantilever on the right-hand side, which is not supposed to move, and the left cantilever, where the myofibril is attached. So once the calcium is released, you can see that myofibril is now contracting, pulling the cantilever, left-hand side of the cantilever toward the glass needle side. So once force reaches to the steady state, then we measure how much this left-hand side cantilever moves due to the muscle contraction. And we calculate this muscle force using displacement of the left-hand side cantilever relative to the right-hand side. You also use stiffness of the cantilever. In this study, we quantify force depression at 1.8 micron of the average sarcomere length. So first, we measures the purely isometric force or reference force shown in the green dot at 1.8 micron. And then we set the average sarcomere length at 2.2, which is longer than 1.8, and then activate it. And during activation, we shorten the sarcomere length actively to the same length as we measure the reference force, which is 1.8 micron again, and see whether this force for both cases are the same or not. The primary outcome for this study is stress. So we measure the force first, and then we divided the force by the cross-sectional area of each myofibril. So this graph shows a representative result as well as the experimental protocol from one sample that we collected. So upper trace shows a stress curve over time, while the lower curve shows average sarcomere length over time. For this green curve over reference force, we set the average sarcomere length at 1.8. And then around this point, we activate the myofibril with calcium and then wait it till the force reaches to the plateau region. Then we have another condition which includes now active shortening shown as red trace. So we initially set the average sarcomere length at 2.2 micron and then introduce the calcium to activate this myofibril. And then here we started active shortening during activation till the final length of the sarcomere becomes 1.8 micron, which is again same as this green line. And then also we waited for a bit till the transient effect of the force is completely gone. As you can see here, even though the stress was measured when the sarcomere length, the final sarcomere lengths were the same, we observed that clear force depression in this myofibril after active shortening. So now let's see our main result. So on the y-axis, stress value is shown as a function of the two different conditions we measured. This green dot shows a reference force that I just showed you in the previous slide. And this red dot means the force after active shortening. White dashed line connecting these two dots mean the measurements were repeated, so the comparison was made within the same myofibril. Pulling all data points, which is 9 myofibril in total for now, we found that the force after active shortening was always lower than the purely isometric force, although again, the comparison was made at the same sarcomere length. So it means that cardiac myofibril do exhibit force depression as we hypothesized. So the next question is what this result means. Since there's no research done in this field of force depression using cardiac myofibrils, I feel it is a bit hard to make a direct comparison to the previous research yet. But I thought maybe I could talk about how our results would be related to the suggested mechanism of force depression. So earlier I mentioned that this one mechanism of force depression, where sarcomere after active shortening would have a lower number of cross-bridge due to this strained actin filaments. So the force is lower than the purely isometric force. Before I explain how our result would be associated with this mechanism, let me first explain about how this mechanism has been supported by other studies. So the first way to address if this mechanism is true or not is to quantify and compare the stiffness of these two sarcomeres. So for example, if we see the structure of this green sarcomere that generates force purely isometrically, the actin filament in this sarcomere provides the binding sites of the myosin heads properly, meaning this part anchored to the myosin filament firmly. And now when we look at this red sarcomere, which is after active shortening, this white part of actin filaments would still provide the binding site for the cross bridge properly, but this strained part, or this gray area of the actin filaments, would not really provide the binding sites well, resulting in this overlap, the actin filament in this overlapped zone would not anchor to the myosin filament fully. So with this assumption on this structural difference, Juma and her colleagues measured how much force is required to stretch both sarcomeres by the same magnitude or measure their stiffness. And they found significantly decreased stiffness in this red sarcomere after active shortening compared to this green one, suggesting that there would be lower number of cross bridge in this sarcomere after active shortening compared to this purely isometric contraction, explaining why we saw lower force in here. The second way to evaluate this mechanism is to investigate energetics. So if I briefly explain about details, in order for one myosin head to finish one cycle of cross bridge, one molecule of adenosine triphosphate, or 1 ATP, is required. And this 1 ATP is hydrolyzed into 1 adenosine diphosphate, or ADP, and inorganic phosphate during the cross-bridge cycle. So it gives us two possible ways to evaluate their energetics. First, maybe we could quantify how many moles of adenosine triphosphate have been taken and used. Or second, how many moles of this one, adenosine diphosphate, were produced for both sarcomeres. Juma and et al. In 2017, they chose the second method, quantifying how many moles of ADP were produced when activating muscle isometrically and after active shortening, to see if there is any difference in terms of energetics. If this suggested mechanism is true, and if there is less or decreased number of cross-bridge after active shortening, we would likely actually see decreased amount of adenosine diphosphates in this red sarcomere. And actually, this was exactly what was found by the team. When they compared the absolute amount of ADP for both cases, they found a significantly decreased amount of ADP after active shortening compared to purely isometric contraction. But also when they normalized this decreased amount, absolute amount of ADP to the decreased force, they actually found no difference compared to this corresponding information of the purely isometric contractions, which further suggests that the force depression could be primarily explained by the decreased energetics after active shortening, which would result from lower number or decreased number of the cross bridge after active shortening. So the last and the most recent evidence related to the mechanism of force depression is the proximity of the myosin heads to the actin filament. So when we zoom in this yellow area or to one myosin head, so now this red bottom is now myosin backbone, and this white filament is actin filaments. So during resting states or when the sarcomere is not activated, the myosin heads tend to close to the myosin backbone. But during activation, myosin heads now tends to close to the actin filaments, generating force. And this proximity of the cross bridge to the actin filaments can be quantified using X-ray reflection methods by comparing the intensity of the X-ray reflection pattern. So again, if this mechanism of force depression is correct, then hypothetically, we would see decreased proximity of the cross bridge to actin filament after active shortening, because not all myosin heads were able to bind to the actin filament. Some of them would firmly attach to the actin filament, but others would just stay here and might be trying to find the binding site of the actin filaments. And this was exactly one of the findings in the previous study published this year, which supports, again, this suggested mechanism of force depression. So going back to the question that I had, how are the results of our study related to the suggested mechanisms? Well, even though this project was a bit of work, considering the duration of the work that I could work on this project, I have a feeling that our study is actually not strong enough yet to support the mechanism of force depression, because we did not specifically measure the number of cross bridge for these two scenarios. However, what we could still say is that when we use different types of muscle that has similar structures of sarcomere, we saw hypothetically expected results that could be supported by this suggested mechanism. So in summary, we found force depression in cardiac myofibrils in this study. And our results suggested that force depression is a common phenomenon that can be observed in both skeletal and cardiac myofibrils, that's where they have similar structure in their sarcomeres. And we also think that a common mechanism is at the origin of the force depression in skeletal and cardiac muscle. That's all I have for today, and thanks for your attention, and I will be happy to take any questions.
Thank you, Seong-Won. Excellent. At this time, I would kindly ask the audience members that if you're comfortable to please turn on your Zoom camera to allow for a better discussion. To ask a question, you can use the raised hand function by clicking on reactions on the bottom of your Zoom screen, and I will call on individuals from there. And if you are unable to use the raise hand function, please feel free to type your question in the chat box and I will read it aloud. Rob, please go ahead.
Seong-Won, I enjoyed that. It was very clear. Thank you very much. In your setup file, in your setup slide, if you could show that as the fiber shortened, it looked to me as if the sarcomeres were all at different lengths. And so it wasn't uniform with all the sarcomeres shortening the same amount. Is that your interpretation of what you showed there? And how does that impact on it? Because normally when we to think about these things, we like to think of, you know, that all the sarcomeres are lining up the same way. But if my interpretation of that slide is correct, some of the sarcomeres were very short and others were not so short.
Yes, actually, that's good point. That's I was actually surprised to see that non-uniformly shortened length of each sarcomeres in that setup as well, because in the literature, many people say that sarcomere length are stable in ascending limb of the force length curve where we are at now. But clearly, when I activate the whole myofibril, not all sarcomeres get shortened. One sarcomere, like a few sarcomeres, shortens much more than the other ones. So I don't know why. First, I don't know why exactly it happens in this length region, but also I'm not exactly sure if this would be the reason of the force depression as well. I know that sarcomere length non-uniformity could be also part of the reason of the force depression, and that's actually another mechanism that has been suggested. But first I tested on the ascending limb, so I'm not fully sure about yet. But one way to clear this idea is we measure the force depression at one sarcomere, so that we are not getting any effects on neighboring sarcomeres, which would be interesting to see.
Yeah, thanks, Seong-Won.
Art, please go ahead.
Thanks. I have
I think we lost
some questions that are, I think, pretty naive. And oh, you're not getting sound?
We are now. Please keep going.
Okay. I have a question about time, and I have maybe pretty naive questions, but basically, I was surprised at how long the time scale was for your cardiac muscle, where it seemed like it takes 10 or 20 seconds to develop force. And I wonder, is that just because something is different about cardiac muscle, or is it because calcium takes forever to diffuse, or basically, why does it take so long to produce force?
Yeah, so I think you're talking about the one of the curve that I showed you, stress over time. That totally depends on how the calcium is released from the tube. One thing that I wanted to avoid is the quick release of solution, because I wanted to just inject the activating solution very much gentle way. So that's why it took like 10 to 20 seconds sometimes to reach the plateau region, which is highly random in my point of view. It could be, so it could be fast if the location where I inject the calcium is close to the myofibril, then it could be really fast. Also, if I release the solution really hard, then it could be also very fast, but I didn't want to lose the testing environment. So I rather have a bit far away from this myofibril, so that the calcium goes gentle and slow way. But it doesn't seem to be really affecting the results.
I see. And the reason I'm interested in time is I'm wondering how long the force depression effect lasts. So I think the plots I usually see is that, say in skeletal muscle, it seems like people do, they produce force very quickly, then they do a little shortening and you see the force come down, and then they'll sustain that for, I don't know, seconds, it seems, and then maybe everything turns off. So then I just wonder, if you just kept going on, say for 20 seconds, 30 seconds, does the force depression stay forever, or does it sort of become normal again?
I actually haven't tested how long I could see the force depressed state in that myofibril system. and the reason that it was a bit difficult for me to test is myofiber itself is very fragile. So once it starts activating and it goes longer than 1 minute, then it naturally loses its force because because of the internal damage, I presume. But for this testing, if I last this setup at least like 80 seconds, and if it is good myofibril, it stays there. And considering it is actually time for the single myofibril, I think it would be a little longer time than the time for the whole muscle level. But that's just my assumption. But I don't know how long it lasts in a whole muscle level, actually.
I see. Well, okay, well, that's good to hear. I'll just tell you what I was expecting, is that I would expect that forced depression should not last forever. Mm-hmm. And of course, what do I know? I'm just making this up. But here, I'll just explain why I thought it might not last forever. So one thing you were talking about is this actin filaments that are, some of the actin filaments are you drew them as wavy, and then some of them are under tension, already under tension, so they're straighter. And then I kind of thought that when you have lots of time, then the cross bridges have plenty of time to sort of detach and then find a new binding site and then attach again. And then I sort of feel like the lowest stress overall would be to equilibrate, meaning that if you had one part that was straight and one part that was wavy, I kind of expect that over time with repeated cross bridge attachments that things would sort of even out. Since I think our expectation is that under normal shortening, things are relatively uniform. The stress within, as you go down the actin filament, I think the expectation is that something is fairly uniform. And then the other thing is the fact you were talking about where the cross bridge can hang out near the binding site or can hang out near the myosin axis. And that also I sort of expect to have some time dependence, where if a cross bridge just bound, then maybe it wants to hang out in that location for a while. But then if you have myosin that was hanging out, or sorry, cross bridges that were hanging out near the axis, then I sort of feel like it had plenty of time now to go grab a binding site, and now it should be in the state where it's close to the actin. And so I'm just wondering what's wrong in that kind of reasoning.
I actually don't know. Just based on the literature that I just read, to me, the strained actin doesn't seem to be, if it is actually really strained, doesn't seem to be able to recover and then provide the proper binding site for the myosin heads, even though myosin heads has a fully available to bind somewhere, but actin is just not recovered. That's just my guess, but that's a good question, I think.
Great, thank you.
I'm gonna jump in with a question from the chat, Seong-Won. What are the implications of your research on heart function?
So yeah, actually, that's one of the most popular questions that I usually get whenever I talk about force suppression or force enhancement, but using cardiac muscle. So I've been actually trying to make connections to my research to the heart function, but I actually failed to see the relationship between our results and heart function. I consider our work is more about the basic research of how muscle contracts and how we could explain the phenomenon that cannot be explained by the cross-bridge theories and classic studies. And the reason why I feel a little difficult to make a connection between our results to the heart function is because of, well, that could be one reason of out of many reasons, is that because of the definition of the force suppression. So when we compare the length of, when we compare the stress after active shortening, we actually waited for a long time till the transient effect on the force is gone, completely gone. So after active shortening, we just waited like 20 seconds, which is not actually happening in a heart. The heart is more like cyclical pumping movement. So it was not able to directly compare that phenomena that we saw in our study compares to the actual heart function. But yeah, it would be really great if I could just work on heart functioning, and maybe in the future I will be able to work further on it.
Thank you. Michael, please go ahead.
Thanks, Seong-Won. That was really nice. I'd reiterate what Rob said. It was really easy to understand, which was great. So I was wondering whether you see similar amounts, magnitudes of force depression in maximal and submaximal contractions.
So I haven't actually tried in this cardiac muscle, but I somehow seem to remember that if we do lower, so sub-maximal activation compared to maximal activation, I think we seem to find decreased amount of force depression compared to maximum activating states, but that's still in skeletal muscle.
Mm-hmm. That's okay. I kind of assume if it's similar in skeletal muscle, it's probably similar in cardiac muscle, like you showed. And then my second question is, what happens if preceding the period of shortening, you have a period of lengthening? So you have force enhancement followed by force depression. Do you have less force depression because you might still have the titin being bound to the actin, or is it that the titin then shortens anyway and reduces any benefit that it would have on the force?
That's a good point. I actually haven't tried. I think that would then give us an idea of how potentially titin, if titin actually interacts with actin, then how that contribution of titin would result in the force depressed states. But one thing that just in my mind is that titin doesn't seem to be, let me say it differently. Force depression seems to be more related to active components, the interaction between actin and myosin heads, whereas force enhancement seems to be more related to other components other than actin and myosin. I personally hope to see it is really related to titin, but I just need more evidence to say that yet. But so yeah, I don't actually know whether putting force enhanced states and then measure force depressed states would actually change the magnitude of the force depression. So.
Yeah, I guess I was just thinking that maybe the titin could provide some sort of structure to the actin if it is bound. So then it would be more, the actin might be more stiff, so it prevents that kind of like disruption, if that's what's happening.
If that's happening, yeah. So also, I'm not sure how, what specific part of actin would actually relate it to titin interaction. So I don't actually know.
That's okay. It's mostly just hypothesizing. So thanks for putting up with my questions.
Marco, please go ahead.
Thank you very much. Thank you, Seong-Won, for the wonderful talk. It was very nice, very clear. I have a few questions. So when you talked about the skeletal muscle, you talked about frog muscle, right? When you showed the force length relationship, correct?
When I talk about the skeletal muscle and show the force length graph, that was actually modified for rabbits, not from frog.
But it wasn't, no, the first time that you showed the force length relationship was from frog muscle, the one from basically Gordon, Huxley, and Julian's, correct?
Yes, so that study, so that they did with the frog muscle, but I modified that graph into rabbit's muscle.
Okay, so then my question is, what is exactly the force length relationship for the rabbit? And if it is similar between skeletal and cardiac muscle, or are they exactly the same?
In the literature, they are supposed to be the same, like a cardiac versus skeletal. If they are within the same species, then it is supposed to be the same. Like the length of the actin filaments and the length of the myosin filaments, as well as length of the Z disc, is supposed to be the same.
Okay, and then if you look at the descending limb of the force-length relationship, is it longer than the one from, let's say, the frog muscles?
I don't specifically remember, but I somehow seem to remember that as the species becomes bigger, it sounded like force length curve, the peak seems to be shifted to the right. So I have a feeling that the descending limb of the rabbit muscle would be shorter than the frog muscle, but also I'm not fully sure whether the whole actin length is actually longer in rabbits compared to the frog muscle.
Okay, then let's suppose that you do your experiment, okay, at different parts of the force-length relationship, all right? And then, but you have the same amount of shortening in both conditions. Okay, let's say that you have 0.2 or 0.4 micrometers of shortening. Okay, but one you're starting a little bit lower in the descending limb, and the other one you're starting a little bit closer, let's say, to the plateau of the force-length relationship. The amount of force depression is the same, or is it dependent on the length where you're basically starting your experiment?
That's a good point. I don't actually know if the changing length range on the force length curve would result in the changes in the magnitude of the force depression. But I have a feeling that measurements, of course I haven't done it, but I have a feeling that that would be greater magnitude of the force depression on the descending limb compared to the ascending limb. And yes, because the reason is because in ascending limb, it starts at shorter length, meaning that according to the figure that I provide, the white area on the actin filament compared to the gray wavy part of the actin filament would be longer at short sarcomere length compared to the whereas the sarcomere length is actually starts at longer length and then shortens the same amount, then the parts of the actin filaments where it already started the interacting with myosin filaments actually short. So by the time the sarcomere becomes shorter, I have a feeling that the force depression might be a little greater, but I haven't quantified it yet.
Okay, and then you mentioned the study that measured, let's say, the change in ADP, correct?
Mm-hmm.
Showing that you have less amount of ADP, which would be basically related with less cross-bridge cycling, correct?
Yeah.
And is that related with how much of overlap you get between the two myofilaments? Let's say you showed a small little piece that you had the cross bridge connected to the thin filament, correct?
Yep.
But if I have now more overlap between the two, does that mean that I have more ADP in this case because I started with more overlap?
Yes.
Or not? Yes.
Yes. So that would be correct understanding according to their findings. So greater overlap would provide greater number of possible cross bridges that could possibly attach to the actin filaments. Then more ADP would be hydrolyzed.
Okay, and then my last question is the range of movement between the thin and thick filaments. How much does that affect force depression? So if I
Yes.
have a small shortening, or if I have a longer shortening, how does that affect the values that you showed us?
Yes, so it was actually found in the literature. So if we shorten much longer length, so if the length changes a lot more than the other one, that this sarcomere will have greater force depressed. So the more length change will result in greater force depression compared to short length range changes.
Okay. Thank you so much. Congratulations. Nice talk.
Walter, please go ahead.
Yeah, thanks, Emily, and thanks, Seong-Won. Really interesting questions, and maybe just to clarify a couple of things about some of the questions. So the descending limb for frog and rabbit and really pretty much all vertebrate species that I know is actually exactly the same length, because it depends on the myosin length, because it doesn't depend on actin. So when you think that you have the myosin there, then you have overlap on this side for half of the myosin length and here of half the myosin length. So twice the half myosin length, so the total myosin length gives you the length of the descending limb, which in vertebrate muscles is about 1.6 microns, and then you subtract the bare zone in the middle, about 0.2 microns. So the descending limb is about 1.4 microns. This is not true for some invertebrates, because they have thick filament lengths that are 4, 5, 6, 7 microns. But frog, cats, mice, all mammals that I know, all vertebrates that I know have essentially the same myosin lengths and therefore the same descending limb length. And then there was a question about the actin lengths in rabbit and frog, and in rabbit it's longer. It's about 1.08, 1.09 microns for the actin, and in frog it's about 0.93 or 0.95, depending on the literature that you read. But the one question that I wanted to come back is I think Michael Bagley asked it about what happens when you have submaximal activation compared to maximal activation. And in cardiac muscle, as far as I know, and Seong-Won was perfectly correct there, that I don't think that's really known. But in skeletal muscle, the amount of force depression seems to be very tightly and almost proportionally related or linearly related to the work performed during the shortening. So if you have less force, then you get less force depression. And another thing that was not brought up, and that's why I wanted to bring it up here, is it also depends on the speed of shortening, because in skeletal muscle, based on the force velocity relationship, when you shorten really fast, then you get very little force, and therefore the work performed is much smaller than when you shorten the same amount at a slow speed. And Seong-Won's experiments were done at a very, 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 very slow speed and with maximal activation. So if cardiac muscle, which we don't know, behaves the same as skeletal muscle, then probably the force enhancement for the amount of shortening that was done there would be maximized. If you had a submaximal activation or you did it faster, then if it behaved the same as skeletal muscle, it would be lower. But the question that I have for you, Seong-Won, is I was looking again, and I was surprised again about the raw data graph that you showed, because in the control activation at 1.8 microns and also in the shortening one that starts at 2.2, it seems that half of the force is passive. And so we know that cardiac muscle has very high passive forces. And if you now think that you have a faster shortening and not always fully activated, then the active force will probably be even smaller, let's say only a quarter of the passive force. And I wonder sometimes, really how, well, I'm sure the active force, of course, is important because the heart doesn't beat anymore when you have just passive force. When the heart doesn't beat. But, you know, I wonder how important really that passive force is. And I would assume it might be much, much more important than in a skeletal muscle, where passive forces in the normal range are usually very small. And I wondered if you had thought a bit about this interaction of the active and passive force in your experiment.
Actually, no, I I haven't really considered of the interaction between passive and active force related to the force depression at all. But
yeah, go ahead.
Yeah, but I mean, one thing that we could just quickly test is maybe start activating it at different development of the passive force. So once we start passively stretch, then due to the stress relaxation curve, there will be increasing moments of the passive force. So maybe we could just not wait till the passive force reaches and then to the steady state. Maybe we could activate at different time points. So the length is there, but the passive force is different over time, so that we could differentiate the timing of the activation. Then we might be able to answer that question and interaction between the passive force and active force.
Yeah, an interesting comment, I think it was, but I think Marco brought it up, was the, you know, stretch shortening cycle, if you had done that, and maybe Venus might know more about that, but I chatted with, you know, some cardiac physiologists about this, because the idea was always, you know, that the blood gets into the heart and the heart gets bigger, and essentially, you know, the sarcomeres and the muscle is stretched passively. But then I remember Henk Kronzie told me that no, it's actually at the very end when it's stretched, it's already activated. But of course, activated EMG does not necessarily mean that force is already there, active force. And I wouldn't know what the electromechanical delay in a cardiac muscle is, but that might be an interesting thing to explore. Is there actually an active stretch phase at the very end of the cardiac cycle? Well, when the heart, you know, is filling up with blood and the muscle is stretched. And I'm not sure if Venus has an answer to that, or
actually I have looked into this, and just from what I see in textbooks, it seems to me that the muscle during activation we would get some stretching, but
so that there is a stretch shortening cycle.
Exactly, at the end of the filling there will be activation while the muscle, the ventricle are still filling. But as Seong-Won said, it's still dynamic. It's not a steady state.
Yeah. And the other thing, and I know we talked about that briefly before, Seong-Won, but is that, you know, you are in the cardiac muscle there from the plateau onto the ascending limb, and usually in skeletal muscle we see we usually see force depression if we do it, you know, maximal shortening amplitude, but you see quite an enormous amount of force depression over, you know, relatively, you know, 0.4 microns is not that big. And do you think that might be specific to the heart muscle, that it's different in the heart and you get more force depression compared to skeletal muscle over the same length range? Or do you think it might have to do with the myofibril preparation? Because I know in myofibrils, we always tend to see more force enhancement.
Sorry, we are talking about force depression or force enhancement?
Yeah, force depression. Yeah, I was just mentioning that in myofibrils, we tend to see quite often more force enhancement as well. And I just wondered if it's the myofibril preparation, or if you think it might be the heart that gives you quite a bit of force depression.
I actually don't know specifically, because I actually also try to directly compare the magnitude of the force depression and as well as force enhancement compared to cardiac versus skeletal. But yes, you're right that I have a feeling that we are seeing actually much greater magnitude of the force depression that compares to what it was expected. But I still do not know why exactly. In order to know that, question, I think we would need to measure the force depression using skeletal muscle, but at least a similar length range at the same preparation. But till then, I actually have no good answer for that.
And just last, kind of trying to explore maybe mechanisms. So, if you're correct and this happens in skeletal muscle and in cardiac muscle, and therefore there must be, presumably a common mechanism. And if you believe this as result in energetic results and diffraction results in skeletal muscle, then it seems that it's just because less cross bridges are attached following the shortening compared to the corresponding isometric contraction. If you were to speculate why that might be, what would you say? Why do you think that might be? What's happening there on the molecular level that just cross bridges are not as happy to attach after shortening compared to an isometric contraction?
I just have really imagine, it's just pure my imagination on it. I have a feeling that after the actin filament in eye band is exposed to the stress, then I have a feeling that there might be just angular distortion of the molecules of the actin filaments, so that the binding sites was supposed to be there, but this angular distortion, like a small tilt of the molecule, would give less chance for the myosin has to bind there. But of course, I do not know really. So I'm just hoping that maybe with a new technique in the future, if I could just label specific molecule repeated in actin filaments and see how that molecular structure is changed before and after the active shortening, we might be able to answer.
And that's been, the kind of the theory kind of since Marshall and Planck in 1979, that you get that stretch of the actin filament and the distortion of the attachment sites. And the only problem that I have is that when people measure actin filament elongation, then they say it's between 0.2 maximally 0.5%. So this actin filament that's 1,000 nanometers goes to 1,002 nanometers, or maximally maybe to 1,005 nanometers for a maximal isometric contraction. And I have a really tough time that this 0.2, 0.3, 0.4% elongation causes such a dramatic change in the orientation of the attachment sites that now all of a sudden 10, 20, 30% less cross bridges are attached. That would mean that cross bridges are incredibly sensitive to the precise orientation and of the attachment site. And somehow I have a really, just purely intuitively, I have absolutely no evidence, but purely intuitively, I have really a hard time accepting that. So I'm always asking that question to people because I'm trying to see if anybody else has a different, kind of a different answer to that. And but it's an interesting problem because nobody really knows. We all seem to agree there is less cross bridges attached after shortening. All the evidence seems to point to that, but why in the heck that would be happening? That I think that's completely unresolved. Yeah. Anyway, I think that's all I have for the moment. If anybody else has a question.
Rob, please go ahead.
Just a really simple question, Seong-Won. I assume when you activate maximally, you've got an excess of calcium, and so that the calcium sensitivity hasn't changed by shortening versus just being isometric. So my guess is you actually have more calcium than you need to fully activate the fiber. So that's not going to be the issue, perhaps.
I'm not sure if I actually understand your question clearly, but
in order to bind between actin and myosin, you need a presence of calcium. So if you have a low concentration of calcium, you'll get a lower cycling rate.
Yep.
So I'm wondering if that has to do with the. I'm trying to imagine a method of getting a lesser force, and so I'm wondering if there's something in the calcium pathway.
That's one thing that I should maybe think of in the future, but I have really no good answer for that. Yeah.
Can I maybe just quickly jump in here?
Yeah.
Yeah. So in the myofibril, yeah, I assume, Seong-Won, you were working at a PCA of 4 or 3.5, or yeah. So you're normally working at full saturation level where you're way at the maximum activation, and so calcium sensitivity would not play a role in her preparation at all. But what you're saying has actually been proposed in a paper by Edman in 1996 in the whatever it's called, the Scandinavian Journal of Physiology or something, Acta Scandinavia Physiologica or something like that. He proposed that in 1996, but in his mind it would be a temporary depression due to the shortening and the calcium sensitivity and the calcium depression that then would go away after a few seconds. But that's kind of disproved, and it goes back to an earlier question by Art. How long does it actually persist, the forced depression? And in a normal preparation, the longest one that I think ever anybody has maintained a contraction, I think that was Tim Leonard and I in cat soleus, where after the shortening, we kept the cat soleus activated for another 30 seconds. And in soleus, you can do that because even over 30 seconds, you only lose about 8 or 10% of force. You don't get that rapid fatigue. And then the forced depression seemed to, well, the control force and the depressed force seem to be declining at the same rate for, after about 5 or 6 or 7 seconds, and then didn't seem to approximate each other anymore. And so it seemed to us that for all practical purposes, the the force depression would be there for a long period of time, at least 30 seconds. Yeah.
Yeah, I was just trying to look for some mechanism of so where to next in finding that, what's the, if it's in cardiac and in skeletal muscle, what's the next experiment to try and separate this to understand?
That's a good question. I actually haven't thought about the future plan, but if you ask me if there is any chance for me to somehow label the specific molecule in actin filaments using specific antibody, and then do the fluorescence and then compare how the pattern looks different, or if the patterns changed before and after active shortening, then I might be able to see, okay, there's a molecular structure, the changes in the molecular structure in an actin filament. But I'm not even sure if there's any specific antibody that I can particularly label a specific moles, whereas leaving other molecules free. But that's my imaginated experiments.
Thanks, Seong-Won.
Siska, please go ahead.
Thank you, Seong-Won. That was a really nice talk. I really enjoyed it. I have a question, which is I was just thinking about this, but while you were answering the question about and saying that, well, in human muscle, in cardiac muscle, actually the contraction is never similar to these situations, right? Because you say it's a dynamic movement, so you never really. And then I was wondering, so what would be the advantage of the cardiac muscle to present this mechanism? So I'm just thinking functionally, like why would cardiac muscle present forced depression? I mean, it's not obviously an advantage because it's something negative, but I'm just thinking why it would also have it if you never see this contraction type in cardiac muscle that would induce forced depression. And then I was wondering if you think that this mechanism is just there because it maybe allows advantages in the other direction, or there's no reason why it would try to not have forced depression. I don't know, I'm just thinking
So let
out
me
loud.
clarify your question. I'm not sure if I understood your question clearly. So
And
your
Seong-Won,
first question.
in the interest of time, if I could just encourage you to give a brief answer for this one.
Oh, okay. I'm not actually fully sure yet if I fully understood your question, but one thing that I could take an advantage of uses of cardiac muscle for this research is I simply thought that if the active components are indeed the reason for the forced depression, then I thought this similar result should be present in other muscle types. That's why I actually wanted to look at cardiac muscle and if it shows forced depression. So that's one of the advantage that I could think of. So we could say that, okay, we also quickly check other muscle types, but we still saw the forced depression when the sarcomere structure is the same, so or almost similar. So
yeah, no, I mean that I understood. I'm just thinking from a functional reason. Like I'm just wondering why there would be depression in cardiac muscle. But yeah, I that's probably something we don't know yet, right? So
Emily, can I have 20 seconds?
Yes.
Rob, ever since people discovered that rate constants in cross bridges are not constant, Roger Cook discovered this super relaxed state of cross bridge, a much slower binding thing. Ever since then, my thinking has been around that maybe for some reason, cross bridges might go into these a more relaxed, a more super relaxed state following shortening, and therefore the attachment rate function becomes much, much slower for whatever reason. And I'm hoping that Venus will tell me that next time she goes to the Argonne facility with the synchrotron, she can actually measure rates of attachment and detachment, and then we'll know better. Thanks, Emily.
Great. That was a great discussion, and I'm sorry to have to cut it off, but Seong-Won, thank you so much for your presentation today, and thank you to all the audience members for their great questions. Next week, we'll be hearing from Siska, and her talk title is Muscle and Tendon Function in Freely Moving Animal Systems. So I hope to see everyone next week, and as a reminder, next week will be our last HPL seminar for this year. So thank you again, everyone.